In this session, we're going to discuss the process and significance of photorespiration. Also, we will cover how it is different from general photosynthesis. So, let's begin. We all know that plants are living organisms and they make their food with the help of photosynthesis. That said, have you ever wondered if or how plants respire? Let's find out. Photorespiration, coined by Decker and Teo in 1959, is the oxidation of photosynthetic intermediates to carbon dioxide in the presence of light, and mainly occurs in the green cells of C3 and CAM plants only. The process starts from the substrate. For the initial biochemical reaction of the photorespiration is the ribulose biphosphate, RUBP, which is then oxidized to give phosphoglycolate and phosphoglycerate by the enzyme Rubisco, RUBP, carboxylase oxygenase. The glycolic acid is formed by the hydrolysis of the phosphoglycolate by the phosphatase in the chloroplasts in the presence of light. The concentration of glycolic acid depends on the concentration of O2 and CO2 in the atmosphere. Hence, we call these C2 pathways, because glycolic acid is the two carbon compounds. The enzyme glycolate oxidase oxidizes glycolic acid into glyoxylic acid and hydrogen peroxide, which are then transported to peroxisomes. Once this occurs, the catalase enzyme in the peroxisomes breaks down the hydrogen peroxide into water and oxygen. Now, the transamination reaction with glutamate converts the glyoxylate to glycine with the help of the glyoxylate aminotransferase enzyme in the peroxisomes. Glycine is then transported to the mitochondria. Further, two molecules of glycine are converted into serine, CO2 and NH3, by the hydroxymethyl transferase enzyme. This liberation of CO2 in the presence of light is known as photorespiration. The serine produced is transported back to the peroxisomes. Finally, glyceric acid is transported out of the peroxisomes to the chloroplasts and is phosphorylated to phosphoglyceric acid. This process is known as the glycolate pathway. Now we shall discuss the significance of photorespiration. Photorespiration is useful in dissipating excess power. For example, the conversion of glycolate to glyoxylate in the light phase uses NADPH2. Glycolate, formed as the end product, protects plants from aerobic oxidation. During photorespiratory metabolism, amino acids like glycine and serine are synthesized and used as the precursors for many metabolites such as proteins, chlorophylls, and nucleotides. The photorespiration process does not produce energy like other respiration processes. The total carbon, which is fixed by the C3 plants, is estimated to be lost in photorespiration. The photorespiration process can be controlled by increasing the CO2 concentration in the atmosphere, but the excess decrease in the CO2 concentration will close the stomata. Thus, this also explains why this process does not occur in C4 plants, because C4 plants have a mechanism that increases the concentration of CO2 at the enzyme site. A lot of us might often get confused between photosynthesis and photorespiration. A significant distinction between the two is that photosynthesis occurs in the presence of carbon dioxide, which reacts with the Rubisco enzyme, whereas photorespiration occurs in the presence of oxygen, which reacts with the Rubisco enzyme, which will result in different products. Summing up, we have learned the whole process of photorespiration, its significance, and how it is different from photosynthesis. We hope that you now have a better understanding of this important process carried out in plants. Thank you.